right. Welcome everyone to today's data science seminar. This is the data science seminar series from the University of Washington eScience Institute. As part of the University of Washington, we acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot Nation. Joining us today is Shachi Mittal, an assistant professor in the Department of Chemical Engineering at the University of Washington, and an adjunct assistant professor, in the Department of Laboratory Medicine and Pathology. Shachi received her Ph.D. from the Department of Bioengineering at University of Illinois Urbana-Champaign in 2019. Her group works at the interface of spectral imaging, cancer biology, pathology, and artificial intelligence, where they align engineering technologies and clinical science to develop systems useful for patient care, particularly for cancer management. She has received several awards and fellowships, such as the Baxter Young Investigator Award and the Big Data Fellowship for Computational Medicine. I'm looking forward to Dr. Mittal's talk, and I would invite her now to share with us some highlights from her research. The title of her talk today is Digital Technologies for Disease Diagnosis, Integrating Imaging and Data Science. Thank you, Shachi.
Yeah, thank you so much, Dave, for having me, and thank you, eScience, for having me today. So today I'll be talking about a little bit about my prior work and then what is happening in the lab currently. Okay, so just so that you're not lost in the entire presentation, these two slides are going to focus predominantly on a few key concepts that I'm going to use throughout the talk. So how does a cancer diagnosis happen in clinics today? So when a patient comes in, the affected part of the tissue is taken out and it is sliced, and each of those slices are placed on a glass slide. Then these slices are interacted with different molecular markers through antigen-antibody interactions. And what that does is that it ends up giving a color which is different for each type of marker present on the sample. Then these stained slides are imaged under a microscope, which are then used by a clinician. In this case, we call them a pathologist, who then comes up with the diagnostic report. Now, the problem with this approach is that because it is observer dependent, there is subjective evaluation, and therefore it is difficult to standardize from one clinician to the other, from one hospital to the other. There are measurement errors coming not only from staining the sample, but also interpreting the images. And there is limited field of view and use of microenvironment, which I'm going to cover on the next slide. So to give you a little primer of what whole slide imaging is, this is where the sample that is stained initially was placed under this microscope about a decade ago, which somebody would physically slide, look under the eyepiece, and then write something. So there was no record of an image taken. A decade ago, there was this whole slide imaging system, or this closed box that you see, that would actually digitally scan these images out. And with that, this presents an opportunity for data scientists to really come and actually use these images and make more standardized pipelines. So what we really need is for microscopists, software engineers, data scientists, pathologists to come together and work on this problem of cancer diagnosis and prognosis. So how would a pathologist actually detect cancer? So cancer is not always of the same types and of same intensity. When the cells are normal, they grow in just one layers. But when you start seeing multiple layers on top of each other, but still only 2 to 3 layers, that is what is called as hyperplasia, or hyper state of growth. This is not cancer yet, but puts a patient at a 2 to 3-fold higher risk of developing cancer. When you start having a little bit more accumulation of these cells on top of each other and different types of mutations, but the mutational load is still low, that is when it is called as dysplasia or atypical hyperplasia, which is when we have 8 to 10-fold higher risk of developing cancer. When we start seeing carcinogenic mutations, but the cancer cells have not crossed the barrier and spread through the lymphatic system to the rest of the cells, it is called as in situ cancer, or inside. The cancer is still inside from where it actually started growing. When the cancer crosses this barrier and then just starts spreading, that is called as invasive cancer, or invading the space. So when these changes are happening in these blue cells here, there are a lot of changes that are happening around these blue cells, which are not cancer cells, but other cells of our microenvironment. And this is where we have known that it's important to understand what's happening with these cells also, because they play a role in how the cancer cells are going to grow and spread further. So what a pathologist does is they get images like these, where you can see that on the left-hand side, there is this in pink, there are stains, which is called as collagen. So this is called as a hemotoxylin and eosin stain. It's an acid-based stain. All these fiber-looking structures are called as collagen, and the cells in purple, which are growing very nicely in one layer contained, these are called as normal cells. But when these cells start growing on top of each other, they are not looking that great anymore, but they are still contained. This is called as in situ cancer or stage 0 cancer. And when you see that there are changes happening in these cells, the pink part of the collagen part of the tissue no longer remains the same. It looks way more crowded than the top row image. And this is because where this be called as that there is alteration of this microenvironment, which in breast cancer is called as desmoplasia. And finally, when the cancer spreads everywhere, we see even more altered environment, and that's advanced cancer with desmoplasia. Okay, so this is a primer of how a pathologist would look at these images. And you can see that there are a lot of structural and textural features that I'm looking at while I was explaining you this. And this is what we can use with machine learning to actually come up with more automated and digital pipelines. The other concept that I'm going to be working with is IR spectroscopic imaging, which is called as infrared imaging. So there what we do is we take tissue blocks from patients, put them in array format, because what that allows us is that each individual circle that you see here is coming from a different patient. And when we throw infrared light of various frequencies, which means thousands of frequencies in our case, on the sample, depending upon what type of molecule was present at that pixel or location of the image, it's going to absorb those thousands of frequencies differently. So every pixel on this image is going to have a different spectral profile or absorption spectra. And each of these peaks gives us some information about lipids, proteins, nucleic acids, carbohydrates, which we know will change when a cell becomes cancer other than being normal. So this is what we can use with machine learning and detect changes between these absorption profiles to come up with a digital pipeline. So how we did that is I said we threw thousands of frequencies. This is a very high resolution scan, which makes each individual circle or each individual patient about 8 gigabytes. And the entire TMA or tissue microarray is 1 terabyte. So the first step is really to reduce the dimensions or our feature set so that we can computationally build models that are way more efficient. So what we do is here we use the information about the biology of the spectral profile and identify only specific bands that are discriminatory of cancer versus no cancer to reduce the size of our data set. Then, because here we are using supervised classification, we collaborate with clinicians to get an initial label for a few pixels belonging to each of these classes. And then we pass it on through a random forest classifier, which I'm not going to talk about in a lot of detail, but I'll be happy to in questions, which then gives an assignment for every single pixel of that image. And then we can compare it with the H&E stained image, which in our case is the ground truth that gives us an estimate of the accuracy of our model. So when I was doing this, I was curious whether we are picking up the same chemistry that these stained images actually are designed to pick up. So what we did was that we took this data and just reformulated that data, applied a color map. So imagine we're just taking linear combination of our features. So now I'm just changing the feature space. and then applying a different color map, which would then give you an image like this. So this is a synthetic image, which looks very similar to the HNA image, suggesting that we are picking up a similar chemistry as compared to stained images. So with this, for this entire tissue microarray, which I told you was 1 terabyte, has 1 million pixels, and we could get a color or a label for every single pixel on this sample. And these are our final labels of the classifier. Malignant means cancer, benign means no cancer. Stroma is just different types of collagens that we are seeing. So moving forward in my talk, if you see a color that is closer to red, that is something that we are unhappy about. And if you see a color that is closer to green, that is something that we are happy about. Okay. So zooming into 4 different patients here, I told you malignant is advanced stage cancer, and you can see that the model identifies all pixels to be malignant. Normal, which is absolutely no cancer at all, the model identifies all cells to belong to this benign category. Oh, I didn't put my laser pointer. Okay. And this hyperplasia, which I said is normal, but is at 2 to 3-fold higher risk of developing cancer, the model says that all these cells have a signature closer to benign, which is what we would expect. But this atypia category, which is about 8 to 10-fold higher risk of developing cancer, the model identifies that the cells are a mixoid signature of advanced cancer and benign, which is something that we had known in the literature, but it was never proven experimentally that we could actually see these mixoid signatures early on when it's not even cancer yet. So to really look at the actual quantitative numbers of accuracy on this model, we use something called as ROC curves, which gives an estimate of the sensitivity and the specificity of the test. Okay. And higher this area under the curve, so a perfect classification would be an area under the curve of 1. So you can see that across all categories, we have about 95% accuracy with respect to the area under the curve. And if we were to use only top 7 features, so this model, we used hundreds and hundreds of features to train this model, but we wanted to make a simpler model, and you'll know why in the next slide. If we just used the top 7, we had more than 90% accuracy on all classes except loose collagen, which was not a very diagnostically important class. So they said that when we did feature selection, we could significantly reduce the model complexity without losing much on the accuracy of the model. Now, the problem with this is I told you that these are very high resolution scan. We throw about thousands of frequencies. Each circle took 3 hours to image. So it's not something that is translatable in a clinical setting, because in a clinical setting, they don't really use tissue microarrays. This is something that is used in a research setting, when in a small sample area, we get a distribution from large patients. But what they actually use is this entire huge surgical sample that is coming from one patient. Okay? And now if this sample we were to image with the previous approach, it would take months for us to just get an image for one patient sample. So then we, based on how we reduced the number of features in our model, we identified what are the frequencies that are really powerful to separate cancer from no cancer. And on the basis of that, we only shooted those discrete frequencies and imaged only those frequencies, giving a 50-fold increase in data acquisition speed. So then in 3 hours, we were able to image this entire sample, which had 4 specimens from the same patient from different locations through our clinical collaborator, where we knew that everything that is circumscribed by a blue dot is cancer and anything that is circumscribed by a green dot is normal. On the second row, we have the infrared image at one particular frequency, which is called as the AMID1, which gives us the information about proteins. And on the third row, the bottom row, we have the image from our model that we had trained previously. And you can see that if you look at just zoom 1, it's able to identify all the cells which were identified by the clinician as cancer, actually as cancer. So we knew that our model is performing well, and we have also kind of reduced the complexity of the model, reduced the time of imaging by using data analysis. Once we did this, I was curious if we can take some of these models and actually transfer them to other types of instruments that are being developed in the lab at that time. So one postdoc in the lab was kind of building this hybrid instrument that combined visible microscopy with infrared spectroscopy. And the reason why we wanted to do that is visible microscopy has a very high limit for the resolution that we can go up to. So we wanted a very high resolution scan and also the information about infrared absorption. So this is a combination of the two. And this is a lot of data, but what I want you to focus on is just column A, column D. Okay, so in column A, we have the H&E image, which was then imaged by this hybrid microscope. And column D is the prediction from the classification model. And in column E, you can see that our area under the curves were about 90% to separate cancer from no cancer, blood, collagen, and everything else, which made us really excited because it kind of showed that this pipeline that we have developed is transferable across instruments and types of imaging systems. Okay. But again, the problem with this was infrared spectroscopy is a novel imaging technology, which right now only exists in engineering labs. It is not yet a clinically approved imaging technology. And I wanted to focus more on clinical translation at that time. So we started building digital models directly on these stained images that clinicians are used to looking at. So here the goal was now to identify textural and spatial features instead of quantitative absorption. So the general pipeline that we followed were to get the ground truth data, which was then cropped for the different classes that we had, which was passed on to a convolutional neural network. And the first step that we did was to build a very simple model using this convolutional neural network, which is separating epithelium, which are basically the cells, from collagen. And why we did that is because to train a convolutional neural network, sometimes you need a lot of training data. So if we had a very complex model to begin with, our burden on getting that annotation data would have been really large. And once we made this simple model with about 100% accuracy almost, we used unsupervised classification to look at different heterogeneous patterns within these individual compartments. So here I'm just showing how the training data looked like. And what we tried to do was we tried to mimic what a clinician or a pathologist would do. We actually made crops at different levels of magnification or zooms and different sizes, because that is what a human eye would do when they're trying to diagnose cancer. So this is the results from the model, where on the bottom row, you have the actual H&E image, and on the top row, we have the digital image from the model, where it's able to separate all epithelial cells in purple, collagen in pink, and all other types of cells like red blood cells, secretions, combined into others. So then we clustered the epithelial cells and the collagen compartment separately. Epithelial, by the way, is where the cancer originates, and collagen is where the microenvironment changes are happening. So we know that both of them are important, but how, we don't know. So then we looked at the inertia of our clusters across these compartments, and we found something really surprising. So if you look at the normalized inertia plots, for the epithelial compartment, the inertia increased with increasing severity of disease. In the collagen compartment, the inertia in our clusters actually decreased with increasing severity of the disease. So our hypothesis is that this is probably happening is because in epithelium, when a cell is becoming cancerous from being normal, there is a lot of disruption of the physiological structures of the tissue, which would mean that we would actually see more or higher inertia. Whereas in the collagen, there is a lot of coagulatory signals because of proteins and other signaling, which would lead to decrease in the inertia across our clusters. So when we use this spatial indicator, a very simple spatial indicator, and we plotted the normalized inertia of epithelium on the y-axis and normalized inertia of the collagen compartment or stroma compartment on the x-axis for these 100 patients, along this dotted decision boundary, we were able to separate the normal and cancer patients with an area under the curve of 90% just by looking at the simple spatial indicator. And interestingly, if you look at the blue dots or the blue circles, those are the ones which is 8 to 10-fold higher risk. And you can see that they kind of span both the first and the fourth quadrant, which would make sense. And we can see that the blue circles, which are actually in the fourth quadrant, are more normal-like. And the blue patients that are in the first quadrant are more advanced cancer-like. So then we can use this method with genetic sequencing to get more idea about risk stratification of these patients. So moving forward, this kind of gave us an idea that probably the models that we are developing have the power for early detection of cancer. And that's where we decided to move into actually building models focused on early detection. And why that is important is there was a recent study which was published where 240 different cancer slides were given to about 200 clinicians across the world. And here in percentages, you were seeing the concordance or percentage agreement between these clinicians. For advanced stage disease, which is where we can do very little to save the patient, the percentage agreement was very high, 96%. For absolutely normal cases, the percentage concordance was not high because it's still 87%. In 13% of the cases, they were confused whether the patient actually is normal or has cancer. DCIS, which is cancer in situ, which is stage 0 cancer, the percentage concordance was 84%, which means that 16%, in 16% of the cases, they were confused whether it's early stage or advanced stage. And atypical, which is not cancer, but 8 to 10-fold higher risk of developing cancer, the percentage agreement was worse than flipping a coin. So this really suggests that we really need to intervene with assistive technologies in this space, because if we look at the survival curves, they follow a completely opposite trend, where the 5-year survival rate at advanced stage disease is very low, but is 100% at early stage disease. So next, we started building deep learning models based on that, where again, we took multiple patients, built a pipeline where it would automatically go in and crop in areas at different levels of zoom, pass it on to a convolutional neural network, which would separate no cancer from early stage cancer. So to train, we used 33 large surgical specimens, giving us about 20,000 images to train, another 9,000 images to fine-tune and calibrate the model, and 9,000 images coming from 11 completely different patients to validate or have an external validation for our model. Now, the biggest challenge that we faced on this project was this work involved data from 5 different clinical sources. And we'll see the data from lab 1 looked like this. The data from lab 2 looked like this. And data from lab 3 looked like this. So there was a lot of interclinical variations. This is the same breast cancer, same type of stain, so exactly the same marker, same processing of the tissue, just different people and different labs were doing it. So the data ended up looking very different, which can sometimes throw off our models or make them less generalizable. So what we did was we developed a pipeline where we actually created synthetic data that would simulate all possible variations that we might see in this stain. Because even if we are working with just 5 clinics, that might not be the boundary conditions of all the variations that we see on this image in the world. So we created millions of such synthetic images, and we chose a contrast that most clinicians were comfortable working with. And then we trained a model that given any of these synthetic image, which is of the same patient, the model would be able to produce back the target image. So given these input images, this deep learning model is now able to actually normalize for these variations in these images. And based on that, our accuracy numbers actually dropped up for our external validation, where I'm reporting this in terms of confusion matrix. So a confusion matrix basically gives us an assessment of the extent of confusion between the classes. So the main diagonal numbers are the true positives, and the off diagonal numbers are the pairwise confusion. Okay. So here is the confusion matrix for our training or calibration data. And you can see that we had about 85% average accuracy across all our classes. So let's look at this number 3% so that we all are comfortable reading a confusion matrix. This says that 3% of the cases that were actually early stage cancer were confused as benign by the model. So this is how we read a confusion matrix. And we had an exact similar distribution for our independent validation data. But this is something that makes us data scientists or engineers happy, but this is not what a clinician is used to looking at. They are used to looking at images. So then we created a visualization pipeline that would take the projections from these models and actually generate it as an image that would be useful for a clinician. So on the top, we have the actual H&E image that was inputted to this model. And on the bottom, we have the output from the model where in red, it's able to highlight all the places where stage 0 cancer was present. Again, on the second patient, there was no stage 0 cancer present. And exactly that is what the model identified for all cells to be green. And similarly for this third case and this fourth case as well. So about with 85% accuracy, we were able to create this pipeline for early stage detection. And then we moved on in my lab to actually start looking at, can we take some of these models and actually detect kidney disease with these models? So again, interestingly, all pathology, whether it's on breast tissue, kidney tissue, colon tissue, lung tissue, happens with the exact same pipeline, with the same challenges of intra-observer variability. So here again, we first did an unsupervised learning to see what are the different heterogeneous components that we are actually able to isolate from these images. So now this image on the left here is a different type of image. It's no longer an H&E image. This is called as a trichome blue image, which stains different components of the kidney tissue. And this is a clustering algorithm output where it's able to highlight 4 different components of this tissue, which then we can segment and actually isolate all the collagen from this tissue and all the epithelial cells from this tissue. And then we can take this to further being segmented as epithelial segmentation and collagen segmentation, where we were able to pick up different signatures in these classes, which gave us confidence that building a supervised model downstream would actually be accurate for some of these classes that we wanted to build a model for. So then we chose these classes, which are again indicative of chronic kidney disease and would help us tell clinicians early on which patient would need a transplant and which patient would not benefit from a transplant. So again, the pipeline is the same. We create the data set, we train the model, we evaluate the accuracy, and we tune back our model. So to just show the training data, Here are some of the hand-drawn annotations which act as training data for our supervised model. I'm not going to go into the specifics of these classes, but these are basically indicative of kidney disease, like glomeruli. These are the ones that actually filter blood from the kidneys. So when they are not functioning properly, that's where we have kidney failure. Inflammation is where we have our immune cells reacting in response to an infection or failure in the kidney. And similarly, we have all these tubules and fibrosis, which is just an altered collagen profile. So again, we created a pipeline of where, because we end up working with hundreds and thousands of images, so we created a pipeline where we can actually, the model can take in, crop out these images automatically and put them in folders assigned to specific classes, which then goes into a convolutional neural network. Our first batch was to use transfer learning to train a VGG16 model. And on the left here, we have the clinical image, and on the right here is the predicted image from the model, where we were able to successfully predict the tubules and separate them from gloms. But again, I just wanted to iterate that message again, that this problem is not only pertain to H&E image, but these are the images that we got from our clinical collaborator, and these are the images that were obtained only from one source, but at different time points over the span of 2 to 5 years, which meant that different lab technicians, different people must have processed these tissue samples, and you can see the amount of color variations or amount of variations that we are seeing with the same marker or biomarker for these images. So then we kind of wanted to investigate if the model is sensitive to some of these changes. So we did a very strong change, which is definitely more than what we were seeing in this data distribution, is we took this region of interest, we perturbed this image to make it look like this altered region of interest. And then we made our model have predictions on both these images. And you can see that the predictions are significantly different, especially for the fibrosis class. And by the way, the prediction on the second image is more accurate than the prediction on the left image. So the accuracy of the model was changing with respect to the variation in the images that we saw. And that's where currently we are working on building a normalization pipeline using cycle GANs, where we can figure out what are the best types of contrast that gives us the best accuracy, so that we can transfer all the images to look similar to those contrast profiles. So now I'm going to switch gears, which is the other component of the lab, is where we are trying to profile our immune response, which is a very important part of the microenvironment. So how does our immune response play a role in cancer development, in response to cancer treatment, and once the patient has gotten the treatment, the probability of cancer coming back? What is the role of immune cells in all of these aspects of cancer development? So here we are using a combination of new imaging and data science. So we are using multispectral imaging system, where we are able to actually tag about 9 to 10 markers on the same slide. So far, where I showed you the images like H&E or trichome blue, these were all 2-color images. We were only able to target at maximum 2 markers. But now we can target about 8 to 10 markers in a high throughput manner with a very high resolution scan, which would look something like this, where different components of the tissue are going to be highlighted. And what we are really highlighting are the immune cells and some of the markers to identify cancer cells. These are all fluorescent imaging, and we use different spectral regions or wavelengths of light to image these different fluorescent markers. So the pipeline that we follow is, first we stain our slide with 8 to 10 markers. We scan it using the system. We annotate to get the data for our model development. We make our models and make a digital report of the patient. So just kind of showing you what the power can be to sample the microenvironment or immune cells. Here on the left, I am showing the H&E image, which was then clustered using our previous deep learning pipelines. And to zoom in, you can see that there are immediately two different populations of collagens that I, with my naked eye, can also see. And the digital model is able to identify and separate these two different spatial populations of collagens. And similarly, here in yellow, the model is able to identify the immune cells, which are at the boundaries of these cancer cells, but they are not actually going in and interacting with the cancer cells. So this is a patient that we would call as actually immunodeficient, in a sense of there is not much of an immune response with the cancer, and there is not a lot of signaling happening between immune cells and the cancer cells, which might mean that this patient may not benefit as much from immunotherapy and some of the other forms of therapy as compared to this patient, where you can see that all of these immune cells, which we also call as TILs, are actually interacting a lot and are dispersed within the cancer cell environment. And this is a combined image where on the left we have the H&E image, and on the right we are able to identify the cancer cells in pink and the immune cells in blue. And you can see that most of the immune cells are again in the collagen, but not so much in the cancer clusters. So it's very important to see where these immune cells are present to be able to understand the signaling that these immune cells are doing with the cancer cells. To show the data how it looks like from this multispectral image, once we can target 8 to 10 markers, is here the first image that you're seeing is where we have the raw data from the instrument, which basically shows you a bunch of colors, but they're all mixed up and jumbled up together. And then we do a pre-processing step, which gives you the second image, which is called as a spectral unmixing image, okay, where we actually get the exact color at each location, which tells us the exact marker that was being absorbed at each location. So in the first row and in the bottom row on the images for the right, I'm basically comparing unsupervised classification with supervised classification, where the top image here is a k-means clustered image using 3 clusters, and you can see that it's kind of separating the boundary of where the tissue is present, where nothing is present, and the individual cells that we see within the tissue. And the supervised classification is able to pick up everything from CK stain, which highlights the membrane of the cancer cells, collagen, PD-L1 are our immune checkpoints that handle and interact with our immune cells, and similarly PD-1. But now when we increase the complexity of clustering, so now I changed the clustering to 6 groups, and immediately we can actually start pulling out the other associations that the supervised classification was able to pick out without having to annotate or train the model in a supervised manner. And another interesting thing is that our supervised classification used this pre-processed image, whereas the k-means clustering actually used our raw data for computation. And again, I'm going to show you another image where the supervised classifier is able to identify the location of different immune cells. In 3 groups, we are only able to see the cancer cells, background, and everything else. But once we start adding more complexity to our unsupervised model, we can start seeing the other immune cells. And this image highly correlates spatially with our supervised model. So to summarize, really what I've presented here are 2 pillars of the research in my lab. One is building artificial intelligence models for medical diagnostics, where we are building deep learning models to identify tumor and microenvironment. Our focus right now is breast cancer, and one of our work is involving kidney disease, but a lot of the pipelines that we develop in the lab are transferable to other diseases. And the second pillar of the lab is to profile the immune system around the cancer cells, both by targeting multiple biomarkers. So that's where we do assay development, we do imaging, and second, complementing that with the analytical backbone, which is where we do image segmentation, cell segmentation, and deep learning. With this, I would like to thank you all. And if you have questions beyond this talk, you can reach out to us. This is my lab where we have undergrads, grad students working on this problem. I would like to thank all our collaborators and mentors and funding sources. And thank you again, eScience, for having me to talk about my research here. And I'd be happy to take any questions.
Thank you, Shachi. That was wonderful. And I'd like to remind our viewers that you can ask your questions through the Q&A button on the bottom of your Zoom screen. And Nicoletta and I can help you share those questions with Shachi. That was really wonderful, Shachi. I wonder if I could ask you a question that's a little less technical and a little bit more high level, because you showed us confusion matrices, and you showed us some really impressive area under the curves. How can you shape, say, decision boundaries between these different cell types? Because I guess I'm wondering that for a patient, the difference between a type 2 and a type 1 error, so a false negative or false positive, for example, it means something rather significant, right? Because we're interacting with patients. So how do you view that? And then what, if anything, can you do when you're sort of choosing or tweaking your decision boundaries or your loss functions to mitigate the substantial potential for outcomes of type 1 and type 2 risks?
Yeah, that's a great question, actually. So one thing is, in the models that I've presented so far, we have not put a different cost to type 1 and type 2 error. They both have exactly the same cost function with respect to the model optimization. And that's because right now, the development of the model was more at the stage of research and model development. But when it comes to deployment, these two errors are going to have a completely different cost depending upon what's the role of the algorithm. So for example, if the role of the algorithm is screening, then a false positive is less crucial than a false negative. Like if you have more false positive, that's okay because you're going to do more testing. But if you miss a patient and they completely go out of the pipeline, that has a huge opportunity cost. So that's where we have to kind of also think about having different costs for these functions, which we haven't had so far. The think about modifying the boundaries, that's where when I used the model for the infrared data, that was a pixel-based model. So when we have pixel-based model, we have less problems with those decision boundaries being very crisp, because especially when you're looking at the spatial distribution, and if I'm having pixels, then you'll have enough neighbors for any annotation that are actually going to belong to the same class. And it's going to be very rare that a pixel of one class is going to be adjacent to the pixel of the second class. But for some of the deep learning models that we built, which was around patch-based approaches, that's where sometimes interfaces become a really huge problem for us, because that's where it's possible that there are some patches which are next to other patches of the other classes. And that's where modifying our decision boundaries becomes very crucial in getting high accuracies on those models.
Awesome, thank you. There's a question from a student. I'm curious if you've considered using cell segmentation instead of labeling pixels. I suspect the training data sets would be more difficult to collect.
Yeah, absolutely. So we would love to, but the biggest problem with cell segmentation is a lot of the cell segmentation methods that exist, they don't have a way to assign an accuracy number, because not the cell segmentation methods, they are totally based on textural feature analysis, and they're not based on annotating different cells and then training the model. So there is no way for us to know the accuracy. So now if you imagine, if we segment our cells and we lose the context of accuracy at that step, then we cannot even assign a confident accuracy towards the last step of our model, which is important for clinicians to believe in our model, one. Second, if we are trying to build complex models, like for example, where we had cancer, no cancer, different types of microenvironments, different types of collagen, that's where we have to then, if we do cell segmentation, then we have to do cell phenotyping, which becomes another problem at another scale. So that's why we kind of chose the route of actually labeling pixels ourselves than having to go with this approach.
Thank you. Let's see, there is another question for you. Early on, I think the initial input size was reported as being on the order of terabytes for one side. After cropping the different zoom levels and sizes, how much reduction do you achieve in terms of data size?
So for the infrared data, where the entire tissue microarray was a terabyte, where we're not really cropping any zooms, but we're actually cropping our dimensionality of the data, that's where a terabyte data actually became about 20 to 30 gigabytes for hundreds of features. And then it becomes 5 to 6 GB, sorry, I didn't mean 20 to 30 terabyte, 20 to 30 gigabytes, and then 5 to 7 gigabytes once we start moving to 7 feature space. So there's a huge reduction in the space. For when we are trying to build deep learning pipelines, there each image, the big image which is stained, is about 2 to 3 gigabytes. And when we crop, we initially actually reduce the data by about 10%, but that doesn't end up being a reduction because then we have to do data augmentation. We have to create so many different copies of that data that it doesn't end up being a huge reduction. But again, that's not so heavily space limited as compared to the infrared data because of dimensionality.
There's another question that's a follow-up that I really like because it gets at my software engineer and my chemical engineer heart. What is the ideal end goal production environment for this? So as a chemical engineer, tell me about scale up, right? Is this meant to be on site, in the clinics, in the cloud? How do you envision the deployment actually looking for this?
Yeah, so the main goal for deployment is actually in the clinics. Once we have tested these algorithms on a very large variety of images and we know that the training is complete, because any clinic is not going to have computational resources to be able to train the algorithm. So we definitely want to work on deployment once the algorithm is packaged in a way that we know that the training is complete and we just have to make predictions. That's where we would like it to be introduced in the pipeline of when clinicians look at these digital images on their computers and they have their own pipeline. They can basically click a button and be like, okay, digital assist on. And then the model will send these projections and say like, okay, you're thinking that there is cancer on this 5% of the image, but the model thinks that there is cancer probably on 50% of this image, which you ended up missing. And that's where they can kind of use it as a second opinion, because we still think that deep learning or AI is still not there where we're going to have a complete automated decision-making process based on these methods. They can be more in assistive roles to the clinicians. That's the end-to-end goal. The immediate goal would be to make it more transferable in a sense where multiple labs can actually work on it in a reproducible manner to optimize it so that the clinical deployment is faster than only one lab working on it.
And I'll ask you, I think one final question. A lot of folks in the seminar aren't ever confronted with dealing with the Federal Drug and the Food and Drug Administration, excuse me. And could you just talk for a minute or two, sort of how you get from a machine learning model to a diagnostic that's been approved by the FDA?
Yeah, So I'm happy to answer this question now because now there are more pathways of where the community or the clinical community considers data science and software development in this field novel enough for them to be called as diagnostic test. A few years from this conversation, that was not even how that was being approached. So last year in November, a company who made a digital model on identifying prostate cancer got their diagnostic test FDA approved. So now there is more and more acceptance to these models actually being useful in the clinic. The two challenges that we have is one, being able to report these AUC values. So like I reported them for 100 patients and they're not happy with 100 patients because there is a lot of heterogeneity from patient to patient. So the first step is really to be able to report these numbers or report ROC curves for a very large cohort of patients. The second is there has to be some equivalent of a clinical trial. Like when there is any panel or new diagnostic test that is developed in a clinic, there is clinical trials that support why that diagnostic test is going to be useful. So then these models need to be deployed in a setting where we have to prove that it helped clinicians come up with better assessments. And that's what exactly that company did for the prostate cancer model. And you have to report how many clinicians, what's the percentage improvement, what's the percentage reduction in the error, did the percentage agreement between the clinicians increased once they start using those models, which kind of becomes an equivalent or proxy to a clinical trial. And the third is that you kind of need the clinical environment to be in a space where adoption of this doesn't add to their time. Because if it adds to their time, it adds to billing. It just puts a whole chain of complex processes in. So like, for example, 5 years before, this would have not been a conversation. Why? Because not every slide is actually imaged and digitized in a clinic. But now there is more and more adoption of, well, we want to scan every single slide. So that's where you are not saying that your pipeline needs a slide to be scanned. You're saying that it already exists. This is just that you're doing something that's going to increase your speed and not reduce your speed. So that's some of the things that we have to kind of think about in terms of deployment.
And I guess it gets just a little bit more complicated as you start to add on some of the sequencing metabolomics for the personalized components. And you have to go through this all again and get an updated sort of diagnostic decision.
Yeah, absolutely. And there, I think clinical trials sometimes even become compulsory.
Yeah.
Yeah.
Seems like if you're using a new metabolic method or a sequencing approach, it probably has to go through that level of scrutiny.
Yeah. Yeah, and this is after you have kind of dealt with the approvals to get samples.
Right.
So getting images and samples and human data in an engineering environment is not that straightforward. You have to go through something called as institutional review board, where you have to kind of outlay every single protocol that you're going to follow on the specimens from now till the end of the study. And then that's approved for you to actually get access to the data, initial raw data for you to do anything. So I think now there is much more communication and engagement in that space, but that is also something that was heavily limiting data science in this space.
Great. Well, thank you, Shachi, and everybody for joining us today. This has been a wonderful talk, and please join us next week for the continuation of the Data Science for Social Good final talks in the series. Thank you, Shachi. Have a wonderful day.
Thank you, Dave, for having me, and a wonderful day to you too.